Good afternoon, ladies and gentlemen. My name is Rajiv Agarwal. I'm the FDA Expert Regulatory Review Scientist.
I think there was some feedback, but it sounds okay now.
Okay. So can you hear me now? Everybody can hear me now?
Yes.
Okay. So I will start.
Yep, go ahead.
All right, very good. Good afternoon, everybody. This is Rajiv Agarwal. I'm the FDA Regulatory Review Scientist, and today we will be talking about the cannabis. So first, I'd like to thank our organizer, Dr. An and Morgan, to extend this opportunity to talk to you guys. So the title of today's presentation is Cannabis, History and Quality Considerations for Clinical Research. First, I like to say what we do in FDA. So my job is to make sure that, and my office job is to make sure that the safe and effective medicine are available and every dose is the same. And this is what we do day in, day out. My main job is to review the oncology applications, but as an expert for the botanical drugs, I also review the botanical application that come for IND or NDA within the FDA. That includes the CDER, CDRH, as well as the other centers, whoever needs any input from the CMC, the quality aspect of any drug. So today we will talk about the cannabis. So before I go in detail, I want to give you an outline of my presentation. So first I will talk the history and definition. It's important to know the history of cannabis, and you will appreciate the few slides that I will share with you how and what was done in the past, in the last century. And we'll talk about cannabis in general and what is the chemistry of cannabinoids that you hear a lot. And last year we issued an FDA guidance to encourage cannabis-related clinical research, and you can find that guidance online. And what are the quality considerations that we look for when we review a investigated new drug application, that's in short IND? And what are the challenges that we face when we review any application, whether it is a research application or it is a commercial application? And what are the future clinical applications that you will see from the different component, different cannabinoids that are found in cannabis? And there are some reference resources that I put together that you can go to and look for if you have any issues, what and how to prepare your application, or what is the important for the FDA to look. So let's talk about the history and definitions. So the first use of medical marijuana comes to United Kingdom by a Dr. William O'Shaughnessy. He was a surgeon in Calcutta during British India and reportedly used the cannabis to give a Queen Victoria for her menstrual cramp. That's how the story goes. Now, in 1840s, the marijuana becomes mainstream medicine in the West, and you will see during the presentation that how it became popular in the West. In 1850, marijuana was introduced to US pharmacopoeia, and the extract of hemp, that's very important. I will describe you the difference between hemp and marijuana, and which was an alcoholic extract of the dried tops of Cannabis sativa, variety indica. Very important for this presentation to remember the word indica. It's a variety that is found only in India, as the name suggests. So Dr. Shaughnessy published a paper in 1839 when he was a surgeon in British India and was also a professor of chemistry in Medical College of Calcutta. Medical College of Calcutta was the first medical college in British India. And he did some research and he found that it has been used in Asia Minor, but there was no documented use of cannabis in the Western medicine. And he used this product for the treatment of tetanus and other convulsive diseases. So from 1850 to 1936, cannabis was used as a primary medicine for more than 100 separate illnesses or diseases in the US. So you pay attention to the bottle, it says cannabis indica, cannabis extract, extract cannabis, and several companies in the United States were actually manufacturing it. Then something happened and the cannabis indica was replaced with cannabis americana, which was the American hemp or marijuana, we don't know. But it was introduced, the extract was introduced in the United States by using the variety that are available in the United States. So far, FDA has not approved any drug that is the extract of a cannabis plant for any medical use. However, the FDA has approved several drugs that you may know that contains individual cannabinoids. In 2018, FDA approved Epidiolex, which was a highly purified cannabidiol from the cannabis sativa, which was marijuana, but not the marijuana itself has not been approved. There are other synthetic molecules were approved, which was Marinol and Syndrose in 1985 and 2016, and a similar synthetic substance that related to THC, Sesmed, was approved in 2006. So these products are related to natural cannabinoids, or they are natural cannabinoid. For example, cannabidiol in Epidiolex.
So Agarwal, it's Morgan. I just wanted to say, it looks like your screen switched back to the double screen view. So if you just click the display settings at the top and then switch to swap presenter view and slideshow, we should be able to get the full screen back.
How's that? No?
Oh, perfect. Thank you very much. Sorry for the interruption.
Okay, so let's talk about the definition. Thank you, Morgan. The term cannabis and marijuana are used interchangeably, but they are not the same. Cannabis refers to all plant product from the plant cannabis sativa, which is an American variety. Marijuana refers to part of or product from the plant cannabis sativa that contains substantial amount of THC. That's very important. And hemp that contain no more than 0.3% THC by weight. So the THC content describes whether it is a hemp or it is a marijuana. So that's very important designation. And recently, a farm bill was published, and you will hear during the presentation in 2018 that describe what is the hemp. So cannabis in general. Let's talk about the difference between the two variety that I talked about, cannabis indica and cannabis sativa. Cannabis indica is the small plant and the cannabis sativa is tall plant. And usually the cannabinoids are the same, but it could be the amount would be different. And that a lot of research has been done over the century that defines what are the exact nature, the composition of these cannabinoids in these two varieties. Now, indica and sativa were introduced in the 18th century to describe different species of cannabis. Hemp described the plant that is found in Europe or Western Eurasia, where it is cultivated for its fiber and seeds. What we call hemp today refers to the industrial non-toxicating variety harvested primarily for fiber seed, and now the CBD. Marijuana, which is the cannabis sativa, gives the high THC content. So this is the summary slide for you to take home message is what is the difference between the marijuana and the hemp. Term indica refers to the variety discovered in India, where it was harvested for its seeds, fiber, and hashish. Hashish is the Arabic term you will see in the Middle East. Charas is the Indian term that is used in Indian subcontinent. These are the, I would call the raisins, and ganja, which is the dry leaves, and the bhang is when you grind the leaves and make a powder mixed with water and ingest it. So that's the different definition that you would see what is popular around the world in terms of what has been used and how it was used in terms of the consumer product or for the medicinal product. Now, hemp is a variety of cannabis. It's a type of plant which contains, I noted before, it's 0.3% of THC. The important aspect is science does not differentiate between hemp and marijuana. The law does. And that's the 2018 Farm Bill that I talked about. Both hemp and marijuana can produce high amount of CBD, the non-intoxicating cannabis compound. However, TC is produced in a very different level. And the next slide that I will show you, it will tell you that the market has been driven by the user. So you will see from 2% to all the way to 26%, and I have reports that you can actually have the marijuana that could contain 30%. I don't know which variety is used in the medical marijuana, but again, that's something for a clinician to decide which is the best component that you can use. I don't know whether 26% of THC will actually help in any way except knock you cold. But again, this is something more research needs to be done. Now, cannabis variety, as I mentioned, contains hundreds of compounds. The exact number is hard to calculate, but the cannabis compounds that we noted in the literature is about 540 chemical substances. Besides THC and CBD, the most popular cannabinoids, more than 100 other cannabinoids have been identified, and they are grouped as a cannabinoids. Now, there are other components in cannabis plants. There are terpenes and flavonoids. While cannabinoids are responsible for the effect of this plant on your body, terpenes are the aromatic molecule which gives each plant its characteristic smell and taste. If somebody is smoking a cigarette and you actually smell terpenes. Cannabinoids don't have any smell. And it has been demonstrated that probably the presence of terpenes in the marijuana modulate the effects of cannabinoids. So it's a very complex group of chemicals that you see in cannabis. But again, more research needs to be done in order to find out if the group of terpenes, which terpenes is responsible to modulate the effect of these cannabinoids. So there's more work needs to be done as we see it's happening as we speak. So let's talk about the chemistry of cannabinoids, how these chemicals are actually formed in the plant. And you can use that synthetic pathway in the lab as well. So GPP, that's a journal. This is found in orange peels, in carrots, is very common terpenes, and oligonucleic acid, which is actually formed from the limonene in the plant. So these two compounds give CBG and THC, CBC, and CBDA. So these are different pathways that are followed within the plant to give the different cannabinoids, and the most popular cannabinoids are THC and CBD. Now, something to remember here, the THCA and the CBDA are actually formed in the plant. THC is not formed in the plant. THCA, when it's formed, because of the UV light or the heat, it gets converted to THC or CBD on the surface of the plant. So once you isolate these two cannabinoids, for example, just to give the reference, you will actually isolate in both the THCA and the CBDA, and then you will have THC and the CBD in that mixture. Now, you will see that you have to make the brownie, you have to put it at 180 degrees centigrade. The chemistry of it is because that temperature converts the THCA into THC. So we'll get the full effect. Or sometimes you will see that during the smoking, you have to smoke the marijuana. So then the THCA convert into THC, or you vaporize it. Then the THCA at 180 degrees, they have a different setting in the machine that will convert the THCA into THC or CBDA into CBD. So this is what you see when it comes out of the smoke. It's mostly the CBD or THC and other acids that are formed during the synthetic process. Now, so what happened? There are three different group of chemicals that you see in the plant that I mentioned, and either they are formed by the natural decarboxylation, means the acid is gone, and then you will get the THC, and then it also produces some degradation products. So I have given you a pathway that gives you CBN A convert to CBN. Then the delta 8 THC is also formed from the THC, which is also present in the plant. And you will be hearing a lot about delta 8 THC. And then you have CBDA, which converts into the CBD by the pathway of decarboxylation, means the acid disappears. And the CBGA goes to CBG, and CBC goes to CBC. CBC further degrades CBL, and the CBLL is also formed, and it converts to CBL. So there's a very complicated synthetic pathway, biosynthetic pathway that is present in the plant. And once you extract it, you are extracting all these components that are actually formed due to the natural biosynthetic pathway, or they are formed due to the degradation by natural degradation by UV heat or oxidation or isomerization. So this thing is happening in the plant that will produce different chemicals. That's what makes it difficult for us to actually gauge that what kind of conditions you are keeping, the manufacturers are keeping the marijuana plant or their products, and how they are degrading, whether and how we need to control all these components in a mixture. So FDA issues the draft guidance in 2020, just last year, and this is what you will see on the when you open the cannabis. We are still working on to finalize the guidance. That was the draft guidance that you will see online. So that was the history and what the plant contains, how the plant process, and what you see in the mixture. So far so good. So I'm sure I gave you an idea that what to expect. Now, what we expect. So the draft guidance covers the sources of cannabis for clinical research. So where are you getting the cannabis from? And what are the quality aspects that we look for? And this draft guidance provide some introduction for what the FDA expect from a research investigator to look for and make the package accordingly. So what we look for? We look for adequate characterization of cannabis, which is your plant, and cannabis-derived compounds. What components are in the mixture? For example, biochemical fingerprint. So what the chemical fingerprint is, once you extract it, put it through HPLC, you will see a different peaks or combination of peaks, or the spectrum that will have different peaks. And once we have that reference, then every product that comes out of it that has to be superimposed is called fingerprinting. And there is a USP chapter, it's called Article of Botanical Origin, and that is Article USP 561. That describes the impurities that are present in the plant, and they are the pesticides. And some pesticides are very unique to the country of origin. Then, for example, if you are getting the cannabis from China or from India, they use different pesticides. We want to know whether you have tested those pesticides. And then the identification of article of botanical origin, there's a USP chapter. Then you have to identify how that plant is. This is the plant. This is exactly this particular cultivar. So these are the tests that are listed in USP chapter. And then we also look for microbial contamination. Depends on what you are going to go for, whether it was an oral tablet or oral solution, or the sterile injection, whatever the method you are using. The USP has different chapters that the sponsors can look for those tests specific to identify the quality attributes. Now, the elemental impurities, which are a lot of these elements are mined from the ground, and the plant actually use those elements for their biosynthetic pathways. For example, green tea sucks up the lead. So some of the green tea preparation, you will see very high lead components, depends where the teas were ground. The same is true with every plant that you look for. So there is a USP chapter that tells you that what impurities that you need to control. So somebody asked me, what are the impurities in a cannabis plant? The cannabis plant would have the impurities that are pesticides, elementals, heavy metals that are known as elemental impurities. And then sometimes you extract with alcohol or hexane. So we want to know how much is the residual left. So these are the impurities that are not present in the plant, but they are introduced through the growing conditions. And you will hear it, what challenges that we see. So then you come to the dosage form. So the dosage form is your tablet or your solution, and there is a different ICH guidances that go through the evaluation and recommendations for the specific dosage form. And if the device is used, for example, the smokes are delivered through the inhaler, then we actually have the different requirements in CFR in order to look for the device, what needed to have in the device. And we usually consult the device to our CDRH part of the FDA. Now, once you make the product, once the product is made, it needs to be contained in a bottle or on a blister. So there is a different ways of looking at the quality aspect of it. For example, most popular excipient or the base, you call it, is the oil. So oil can extract different components from the plastic. So the idea is to use the glass bottles, but the glass contains arsenic. So all these things are controlled, and you will find those information in the USP journal chapters. And we have a guidance that was published in 1999 for what to look for when you are qualifying the container closure that house your product. And then there are specific guidances that relates with the different dosage forms. If you what you need to look for, if it is a tablet or a capsule or injection, etc., or even nasal spray. So there's different guidances, and USP covers those dosage forms. Now, I mentioned the Epidiolex is the highly purified drug substance, active pharmaceutical ingredient. So that's the API, which is highly purified. And basically what happened, it becomes similar in the structure to the synthetic molecule. So these things are important, but the impurity profile that you will see in the highly purified, say, cannabidiol coming from the plant will be different if you synthesize the molecule using a synthetic pathway, and that will generate different impurities. So those impurities that I mentioned in the plant are the pesticides, heavy metals, sometimes aflatoxin or solvents. But when you talk about the molecule that is coming from a synthetic process, that is a different pathway that produces different impurities because you're using different reagent, different solvents, different catalysts. So we control those as well. And that will also produce different degradation pathway because those impurities will degrade differently. So that gets more complicated as we review the quality of the product coming from the highly purified molecule or from the synthetic molecule. So what challenges that FDA chemists will have on a day-to-day basis when we review a botanical application very specific to cannabis? So this is the slide that I prepared that will give you an idea what to look for. And I mentioned that one in brief in the presentation already. So is it hemp or marijuana? That has to be very clearly defined, and this is defined by the amount of THC, how much it contains. Name of the plant. Okay, it's a cannabis sativa. We know that. Now, it is the whole plant or the flowers. Now, that defines the chemical component differently because the whole plant will have a different profile versus the flowers. Then, what are the growing conditions? Where exactly you are getting it from? So what happened during the life cycle of a clinical trial, you want to have the same product. So totality of the evidence is the word that I commonly use nowadays. So if you're growing in say North Carolina or say Kentucky, you're growing, then probably in a particular farm, make sure that the product that you are getting it actually is grown at the same place. Also, when you are harvesting, is harvesting at the same time. So the composition of the components that are present in the plant remain constant because of the circadian cycle, the components varies, depends when you actually pick it. The greatest example is jasmine flower. You will not smell jasmine in the flower, but you will smell on the full moon at night. It's beautiful. I have a couple of plants in my backyard. During summer, they just you don't smell them. So it depends on when you pick them, where you pick them from, that defines the chemical composition. The other word you're going to hear in the regulatory filings and other places, certificate of analysis. People ask me, what is the certificate of analysis? Certificate of analysis is basically a piece of paper where you identify how much of THC you have, CBD you have, terpenes you have, flavonoids you have, how much impurities you have, and impurities I discussed, for example, plant, and what you have in the final drug product. So there's a two certificate of analysis. One is for the extract, for example, and one is for your product, for example, a tablet. So these are the certificate of analysis that we look for. Now, over the last 10 years, that has been a huge interest in order to avoid the organic solvents. So what they are using the supercritical carbon dioxide is a fantastic technique. What it does, it actually extract only the lipophilic molecules, which means that the chlorophyll, the green stuff, will not be extracted. So the only thing you will see is the light yellow extract at the end, which is basically will have wax and other carbohydrates, which are not water soluble, but are fat soluble. So that's a good technique that people are using right now. And the impurities I talked about, the pesticides, aflatoxin, solvents, microbes, mycotoxin, etc., from the plant sources. Now, the water activity in marijuana for inhalation is very, very important. It has to be dry enough so the mold won't grow. And the people do research that, okay, 2% of the water is good enough. For example, you have to have enough water if you want to roll a cigarette. So these are things that needs to be controlled. And how you store marijuana, cigarettes or vaping material in a freezer, refrigerator, or a room temperature. Freezer appears to be a right choice, but then you have to take it out to thaw it. And if you leave it for a long time, it could start developing mold. So these are the things that are the quality consideration that are in place, and you will see in the guidance briefly mentioned there that how to control those things. So shipping conditions to clinical site or storage by patient, very, very important. It has to be very well defined how you store those cigarettes, because these are the natural substances. If you keep your bread in the fridge for a long time, it will develop mold. The marijuana is no different. So it needs to be controlled of how long you need to keep it, when to finish your supply. So these are the importance for the patient safety as well as the quality, ultimate quality of the product. Then you will see a lot of different terminology has been introduced. American Herbal Association published a paper lexicon, and they defined what the broad spectrum, full spectrum company. It's a headache because every manufacturer is trying to capture the market, and they using different nomenclature. what to use, a broad spectrum, full spectrum. For an extract, I will call it native extract that is pure, is coming from the plant. But then a lot of different things are done to the extract that makes a different profile, different combination, which is not present in the plant originally that got created. So that makes it different. But they are all very good. I mean, as long as we have a certificate of analysis, the fourth bullet, that what you have in the extract, that's more important. So these are the challenges that we face. Now, as I mentioned, native cannabis extract, that didn't do anything, you just extracted it with the solvent or with the carbon dioxide. So how does it work? So chemical identification of all cannabinoids and terpenes may not be feasible. It's impossible to identify 540 cannabinoids and other terpenes in your mixture, but the effort should be big. And what we look for is the whole extract is active. If you're looking for the extract, not just the CBD, not just the THC, I mentioned the terpenes modulate the effect. So that is part of the what I call so-called fingerprint. You have to use different analytical techniques to capture those. So we different set of fingerprints. For example, one for the cannabinoids, one for the terpenes, or maybe the analytical methods are growing at a warp speed that we probably will have a method that will cover both of these components and the flavonoids and other components. So the whole FDA consider the whole extract as active, but in your certificate of analysis, we want to see what exactly is the composition. So there's a different fingerprints that I mentioned. Spectroscopic or chromatographic fingerprints are very popular nowadays in order to compare batch-to-batch consistency, which is the fourth bullet. So in the batch-to-batch consistency, there's another aspect for it is the botanical raw material I mentioned, where you're getting it from, and each batch is the same, coming from the same field, picked at the same time, picked and processed a specific part. It has to be properly controlled. You can't use the plant at one point and then leaves the other point. So the cannabinoid composition and the profile will be different. So I read a paper recently where they said most of the cannabinoid stays at the top of the plant and the bottom of the plant. So see if you collect something from the top of the plant in phase 1 clinical trial, and the second phase 2 clinical trial, if it's coming from the bottom, the profile will be different. But all these things will be addressed in the certificate of analysis, which is very important for a quality control of any product. It doesn't have to be just cannabis, even a single molecule that we want to see the profile in the certificate of analysis. So the question that I always ask, so what are the applications currently that we have in the world that uses the different components of the cannabis plants? What have been done? Means how much are in the clinical trial, how much, what components have been studied for what indication. So I borrowed this slide from the Trends in Pharmaceutical Science, and you will see there is a lot of clinical indications. For example, CBD, CBDV, which is one of the component in the plant, and then you have the acid, which is delta-9 THCA, which converts into the THC, and then you have CBC I mentioned, which is one of the biosynthetic pathway, also a degradation product, and CBG, which is again a product of the plant, and also could be a degradation product. They are all present in the plant. And then you have the THCV, which is again a very important component and have been, as you can see in this slide, being used used in different studies that I noticed in the literature. So this is in nutshell what the cannabis is all about. So what I mentioned so far is the history, how it came about. The first clinical trial was carried out in India in tetanus and rabies and epilepsy. And epilepsy was one of the most common indication that I noticed that Dr. Shaughnessy used it for. It's very much used in India even today. And then we have approved a CBD highly purified from marijuana for one of the epilepsy indication. And then, you know, there may be more application coming into the FDA in future, I hope. And you guys are the expert in the field and maybe putting in some application in as a investigative new drug application for different indication. And we will look forward to those applications. But if the main thing is that the quality aspect is the no and no go and no go. So once you look at the quality consideration, I think the manufacturer or the supplier should have a clear idea what are the expectation, and that's why we have written the cannabis guidance. But I wrote another guidance, is a botanical drug development guidance, which I call it master guidance. In 2016, it was published. The first botanical guidance was actually published in 2004, which I have updated in 2016. So that gives you an idea that in a nutshell, what is required for a botanical application, because marijuana, whether it's a hemp or marijuana, is a botanical preparation. And if it is highly purified, then actually there are different guidances. For example, if it's a highly purified, like 97, 98% THC or CBD, then different set of guidances kicks in, ICH Q3A, Q3Bs for the drug product. So there are different regulatory pathways if you were going for the botanical or going for highly purified or going for a synthetic. So different guidances covers different quality aspect of it. Here, I'm just giving an example. If it is coming from a purely botanical source, whether it's a highly purified of botanical origin or it is a botanical itself. So there is a clear definition, regulatory definition. If it is a botanical, it's a botanical, means it is just the extract. But if you purify any of these components that you see within the circle, then it's a highly purified cannabinoid of a botanical origin. And if it is highly if it's a synthetic molecule, then of course it's a single molecule by the synthetic pathways. So there are some online FDA resources that I put together, and you will and I gave the reference to different online links, and you can go and look at it. And the third bullet is the Master Botanical Guidance, which was published in December 2016, and the last one is the quality conservation guidance that we published in 2020. So people ask me, so where do I get the marijuana for the clinical studies? So there is only one FDA NIDA approved site, which is in University of Mississippi, which is the only domestically federally legal source of cannabis for clinical research at this time, but we may expect more of them. And this is they grow indoors, and this is the picture of me next to the plants that are grown indoors, and they have a 2-acre site in University of Mississippi that grow marijuana, and they actually control all aspects of the product, and we are familiar with that product, so it's easy to look at their drug master files and cross-reference the product, and it's easy to do that. But again, the hemp, you probably have to get other suppliers to provide that. There is no FDA blessed supplier as far as I know currently. So that was the total presentation, and I think I kept myself within the 40 minutes time slot for this presentation. And if you have any questions, Please ask. Thank you so much.
Thank you. As he said, please ask your questions either live by unmuting, or you can post in the chat and we can read them aloud.
The question is, has FDA ever approved any whole botanical or plant extract for any, hold on, let me see, for any medical indication, not highly purified? If so, which? So far, FDA has approved 2 botanical NDA. One was in year 2006 for the extract from the green tea, which was not pure extract, which was actually semi-purified, and it was for the genital wart. The second new drug application was approved in 2012 that was for AIDS-related diarrhea, I believe, and that was also not native extract, but it was semi-purified, which was crofarabar. So the highly purified one was the Epidiolex that I mentioned. Now, how can FDA actually change its approach to approaching such agent? I don't know. We are learning as we are growing into the understanding of what to do and how to review application, but we have all the resources that I need in order to look at all the application that comes to the FDA. We have highly dedicated staff, very well trained in order to handle any botanical, whether it is coming as a pure extract or semi-purified extract. We published a couple of papers in Science and Nature over the years. You can access that just by typing my name, and that will give you an idea what we have done so far as the approval of the botanicals are concerned, and how many applications we have, how many in each clinical divisions. So that was published, I believe, last year in Journal of Natural Product. You can access that journal online if you just type my name and Journal of Natural Product, and the botanical and these strains will take you to that journal article. The question is, can you explain the regulation behind marijuana dispensary where CBD products are sold with the marketing for relaxation and other seemingly medical indication? I don't know how to answer this question. The CBD products became legal after the publication of the Farm Bill of 2018. You can imagine people are coming with different indications, CBD, different components, not components, CBD is a component, but different analogs of CBD, and they are selling in the market for different indications. For example, as you mentioned, relaxation. We haven't seen those yet in FDA, but once it comes for evaluation, then we will evaluate whether it actually indeed provide relaxation or not. So that will be a controlled clinical trial, and we will look at those products once we receive and review it. this is a long question. Part of the problem seems to be, I'm reading the question, that it is all dependent on proprietary use. This reduces global access to many agents, yet most people rely on these type of agents. The corruption that patents and profits endangered has placed lives in jeopardy the world over. People have used cannabis for many thousand and others in Siddha and Ayurvedic and Tibetan Chinese traditions from around the world. FDA seems to ally too often with industry and not enough with patients. I don't know how to answer this question. I've been in FDA for a quarter a century. I think we look at the evidence, we look at the data, and then we comment on it. So the patient safety is the first. We take that job very seriously when we review any application. There is absolutely nothing that we don't do to make sure that what we review. So the people talk about a lot outside that it used for this and it will cure you for this. And but again, unless we review the evidence, it's really hard to say that what is the duration, what dose you should use safely and effectively. So that is part of the review process. People are using it for years and years, thousands of years. Marijuana or cannabis, hemp products are available around the world for centuries. I think the first known use of cannabis was 5,000 years ago in China. So yes, it has been used, but I think when it comes to a drug that you will use with full confidence, and I think we need to see the evidence and the data as they come in for our evaluation.
Sorry.
The question is, evidence is important. Is the barrier too high to obtain approval? No, I mean, the most important thing that you have to present the evidence. I sometimes give an example to my wife, when my son was young, dress him so nicely that people will say he's cute. So depends how the data is presented, what evidence are there. Are the convincing evidence within the application? So that's pretty much is the job that we take very seriously, that what is provided and how it actually look in terms of the safety and the efficacy. My job is to review at the quality part of it, where what is in the bottom, and that is my day job. And I make sure that everything that is in the bottle that the patient sees is of high quality. And our medical officers, they look at the evidence. Our pharmacologists and toxicologists look at if it is safe, no deleterious effect or anything that is poisonous intentionally or non-intentionally is not there. Or if it's there, how much is there, and there are different guidances to deal with it. So it's a very fulfilling process to see the application. My wife asks me all the time, what do you do? I said, I just make sure that whatever drugs you take is safe and effective. For my job, it's safe.
Yes, just somebody else say something. Well, if that's it, maybe that.
Anybody else have any questions? Well, thank you so much, Dr. Agarwal. That was an incredible presentation. We thank you for your time. We thank you for everybody joining us here today. And we hope that you enjoyed the rest of your week, the rest of your weekend. and everyone just have a good day. Thank you.
Thank you, Margaret. It was a pleasure to be here talking to you guys, and have a great week and the weekend too.
Bye, everybody. Thank you.
Thank you.